Good afternoon, everyone. My name is Natalie, and I'm a PhD student at Queen's University in Dr. Jagdeep Walia's lab. Today, I'll be discussing the dose response for a gene therapy treatment for AAV variant GM2 gangliosidosis using an adeno-associated virus. I will first start off by discussing the biochemical background of GM2 gangliosidosis. So the hydrolysis of GM2 ganglioside occurs within a cell's lysosome and requires specific synthesis, processing, and combination of three gene products, Hex A, Hex B, and GM2A. However, mutations in any of these genes result in a deficiency of hexosaminidase A, or Hex A, or GM2 activator protein, which form a complex to mediate the breakdown of GM2 gangliosides. My project focuses specifically on AB variant GM2 gangliosidosis, or ABGM2, which occurs when there's a mutation in the GM2A gene, resulting in an inability to produce a functional GM2A protein. This leads to pathological buildup of GM2 ganglioside within the cell's lysosome, resulting in widespread neurodegeneration and death by age 4 in its human infantile form. ABGM2 is a monogenic disease, making gene therapy a plausible and likely effective method of treatment. I'll now turn your attention to the blue box in the bottom to go through the principles behind our proposed gene therapy. So by encapsulating the otherwise mutated gene, GM2A, into a viral vector, in this case the adeno-associated viral vector serotype 9, or AAV, we can then administer that to patients that are affected by ABGM2A. On a cellular level, the virus will release the genes into the cells, where the gene will then travel to the nucleus and eventually result in GM2A protein expression. To test the efficacy of gene therapy for ABGM2, I conducted studies on GM2A knockout mouse models. The GM2A knockout mouse model is phenotypically indistinguishable from a wild-type mouse. However, GM2 gangliosides moderately accumulate starting around 20 weeks of age, allowing us to assess the biochemical effects of this treatment. At 5 weeks of age, I began orally gavaging immunosuppressants rapamycin and prednisone. A week later, at 6 weeks, I performed intrathecal injections of the gene therapy. At 8 weeks, I began monthly behavioral testing and blood collections. The short-term endpoint for this project was 20 weeks of age, of which all of the mice were euthanized, and I collected blood and organs. Later on, I conducted biochemical and histological analysis. The box at the bottom outlines the cohorts involved in this study, of which there are 3 different doses that I administered. I'll now briefly discuss some of the results that I have on this current study. So I'll draw your attention first to the graph titled GM2 ganglioside accumulation in the midsection of the brain. Here we can see, unexpectedly, that the medium dose was the only cohort that had significantly reduced GM2 accumulation within the midsection of the brain. And this correlates with the biodistribution in the midsection of the brain as well, the graph right underneath it, where we see the highest expression of GM2A within the midsection of the brain for the medium dose. We then can look at one of the behavioral parameters that we used to assess the phenotype of these mice, and we see that there is no difference between the cohorts. However, this was expected because, as previously mentioned, GM2A knockout mouse models are phenotypically indistinguishable from their wild-type littermates. So in conclusion, the medium dose obtained the best results in regard to the highest reduction in GM2 ganglioside accumulation and gene expression. We had hypothesized that the high dose would be the most efficacious, so in order to rule out human error in the high-dose injections, these cohorts will be re-injected and reassessed. We also saw that almost 10% of the cells in the midsection of the brain were transduced, which could provide a correctional effect of ABGM2. Future directions involve creating a more phenotypically representative mouse model of the human ABGM2. The moderate phenotype observed in GM2A knockout mouse models are likely attributed to an alternative sialidase pathway that also functions in the breakdown of GM2 ganglioside. Therefore, future experiments include creating and characterizing a double knockout mouse model for the GM2A and NU3 gene. Thank you everyone for taking the time to listen to my presentation. I would like to thank my entire lab and my wonderful supervisor, Dr. Jagdeep Walia, for assisting me in this project, as it would not have been possible without them. If I don't have time to address your questions, please feel free to email me. I have posted my email at the top of the slide.